Welcome back. We are starting with chapter 32 and we're going to discuss about the innate host resistance. This chapter is also sometimes known as the innate immunity or your own defense mechanism that works against a variety of microbes in the first place. So let's take a look what we are going to talk about. The first thing that we are going to talk about is the immunity arises from an innate resistance and adaptive defenses. Now, the adaptive defenses, we are going to talk about that in chapter 33. This is your first line of defense, and we are going to talk about that how immediately, non-specifically, this particular defense system works. This is not specific to any organism, and this basically keeps us safe 24/7 every day. So first, we need to define some of these terms. So the most pathogen, which are the disease-causing microbes, must overcome the surface barriers and reach the underlying tissue. To overcome the host defenses, which is innate resistant mechanism that we are only going to talk about in this chapter, and also the adaptive immune response that we are going to talk about in the next chapter. When most of the pathogen overcome these host defense mechanism, that can cause an harm or an onset of a disease. So what is the definition of an immune system is next. The immune system is composed of widely distributed cells, tissues, and organs, and they can recognize foreign substances or microbes and acts to neutralize and destroy them. Also, on the other hand, the immune system can also work towards an injury that is sustained by the cells, either internally or from an external source. The general definition, on the other hand, for the immunity is the ability of the host to resist an infection or disease. So when we say that a person is a little bit of immunocompromised, that means the person's immune function is a little bit compromised, and chances of getting a disease or invasion of the pathogens are a little bit more for that particular person. That means that person is more susceptible to a kind of infection with a diminished kind of immunity. And that can cause due to a variety of reasons we are going to talk about elsewhere. Immunology is basically the study of the immune response and how they protect the host, in this case, the human beings or us. So the first we are going to talk about is the innate immunity, and we call this one as a first line of defense. And this offers the resistance to many microbe and the foreign material, which could be detrimental to our cells. And these includes general mechanism like the presence of a skin, presence of a mucus to all the bodily opening that are membrane bound. There is always a layer of mucus which is moist on them. And also lysozyme, we're going to talk about that. And these lacks immunological memory. What we mean by that, the innate immunity or the first line of defense don't distinguish or remember whether a Staphylococcus aureus tried to infect you, or a Klebsiella tried to infect you, or a virus or a fungus tried to infect you. They work regardless of what kind of pathogen they are in a very generalized basis. Now, the pathogens have to overcome these barriers before they can go into the second or third line of defenses. On the other hand, the adaptive immune response is activated by the cells and chemicals of the innate immunity. And the resistance is tailored to a particular foreign agent. So what we mean by that, activated by the cells or chemicals of innate immunity. So the first is that your innate immunity do contain a first line of defense, which are physical or chemical barriers. Once the physical and the chemical barriers are breached in your innate immunity, there is a second line of defense, which is also inside the innate immunity, which are some specific cells which are attracted by some of the chemicals that are released from the site of infection. And these cells will process the event or the invasion of the pathogen, and they will release certain chemicals which will activate the adaptive immune response. And the resistance is tailored to a particular foreign agent. Even if you are just infected by a bacterial species, again, in between different strains, your adaptive immune response can distinguish in between the strains and could be very, very specific. And also they contain a memory. And that's the reason that the vaccination work. or usually if you get a disease, you do not get the disease once again later in their lifetime. So what we mean by this, that the memory means that some of the cells in your immune system, which are the part of your adaptive immune response, are going to recognize these pathogens, and they will immediately activate the system once they see that pathogen once again. Now, there are, of course, a lot of exceptions of that. The pathogens do have the ability to bypass this mechanism. Say, for example, we can get a common cold many times in a year, but usually if we just get a chicken pox, it's only one time lifetime event. We are not talking about the shingles, you can get that much later, but usually many of these viruses can affect you only at one time. And that's one of the reasons that as a child, we just do get these vaccinations that protect us lifelong. And this is due to the memory of the adaptive immune system. We are going to talk about that in the next chapter. So some definitions. What are antigens? So the antigens are recognized by the immune system, and the immune system can differentiate between a self-antigen or a non-self foreign antigen. So first of all, what is an antigen? An antigen is usually a protein or a glycoprotein or sometimes a carbohydrate complex which can be recognized by your immune system. Most of the times they are limited to the proteins. On the other hand, the lipids, although recognized by many of the structure in your immune systems, are not considered as an antigen. So to qualify for the antigen, most of the times they have to have a specific three-dimensional structure which doesn't change over the period of time, such as a protein, which does have a specific three-dimensional structure, or a glycoprotein, or sometimes some sugars or complex carbohydrates, which does have a very specific three-dimensional structure. Those are considered as antigens. But sometimes there are smaller molecules can act as an antigen and can amplify the immune system reaction. We are going to talk about them in a separate discussion. So the antigen enables the immune system to get rid of the host of foreign invaders as well as eliminate the cancer cells. What we mean by this, the first thing is that each of the foreign pathogen will have a specific antigen that is not present in the host cell, and they can be recognized and can get rid of. On the other hand, the cancer cells sometimes express some of the protein molecules in an abnormal condition on their surface, and these can be recognized by your immune system to eliminate these cancer cells. And obviously there are many cases where this is not possible. There are always a way to bypass that. The pathogens can evade this system, even the cancer cells can evade that system as well. And when this ability is lost, it is the basis of an allergic response or autoimmune disease. So what we mean by the second thing is that the immune system can differentiate between self-antigen and non-self or foreign antigen. In our body, we have these thousands and thousands of different proteins and the glycoproteins. And our immune system do understand which are the molecules or the antigens are self. That means these are the molecules present in the host or the human being. And they can distinguish the ones which are not present in the human body, which are non-self, as foreign. But sometimes this ability is lost, means our own immune system cannot sometimes recognize our own proteins, and this can lead to allergic response, and sometimes the most important one can lead to autoimmune disease like psoriasis or rheumatoid arthritis. These are some of just a few of these autoimmune disease where your own body cells are attacked by your own immune cells as they do not recognize your self-antigen. So the next thing that we are starting is the innate resistance start with the barriers. And what we mean by this. So the first thing is the pathogen must have to breach some of these barriers before they can gain access to our sterilized body compartment. And along with the host flushing mechanism, barrier are the first line of defense against the microbes. So let's take a look at what are these. Say for an example, the sebaceous gland produce the sebum, which do contain some of the specific fatty acids they contain lipocalin, and these are antimicrobial. And these sebaceous glands secrete these sebum all over our body surfaces, particularly in the facial tissues. They are much more active. On the other hand, along with that, the tears do contain a particular enzyme called lysozyme, and we're going to talk detail about that. Along with that, there are waxy material which are secreted by the epithelial cells, which makes a barrier. Our top 30 to 50 layers of cells are dead epithelial cells, which are called as keratinocytes. These are impervious to most of the microbes and make a barrier, or skin is a first barrier. Although you can argue that there are sweat pores and dermal pores through which the pathogens can invade deeper into the tissue, that is correct, but there is always this defense mechanism. Although the saliva do contain this lysozyme, also along with that, they contain a molecule like lactoferrin. Lactoferrin is similar to transferrin, which is used to take up iron from our intestine. On the other hand, lactoferrin is present, or they are released in different types of mucus secretions, which limits the access of iron to these pathogens. Iron is necessary for these pathogens to survive. On the other hand, peroxidase is also another enzyme which do produce some of these reactive oxygen species to kill some of the pathogens. We do have these cilia in our lower respiratory tract, and that is also another source to trap the microorganisms. Also, along with that, the low pH of the sweat, stomach acid, normal microbiota or microbial flora of our gastrointestinal tract, intestinal enzymes, these are all different barriers of the innate resistance. Say, for example, one of the very specific ones is the Paneth cells do produce these lysozymes and cryptin molecules, and these are very specific antimicrobial enzymes and proteins. Along with that, the process of defecation and the urination also flush out microbe from our system as well. So along with that, the tears, as I said, also flush out microorganisms from our body surface. So these are the barriers in innate resistance overall, and we're going to talk about a little bit more detail about these. This diagram just shows a stratified cornified epithelial cells. And what we mean by this, that the top layer of the skin is called the epidermis, and the epidermal layer contain several different layers which are not needed, but just saying that they arise from the stratum basale, which are the actively dividing layer. Then you have the stratum spinosum, where the cells are being matured. Then the stratum granulosum is the last layer, that is the living, and the cells are getting flattened at this point. And then you have the stratum corneum, which are the dead cells. And these cells do contain a large amount of keratin, which is the abrasion-resistant protein, and they also extrude lipid or wax, which is present in between the cells. These are the natural barrier towards the microbes. And the associated microbiota, which are present naturally on our skin surface, also do have some antimicrobial activity. And what has been just pointed out, and we're going to come back to that later, that here is a cell which is called the dendritic cells. These are some of the cells which are migratory cells and move around in the epidermis and look for pathogen. And if they find these pathogens in this epidermal layer, they will alert the immune system. These are the deeper layer, which is the dermal layer, and then hypodermal layer. These are devoid of pathogens. If there is a breach of the first layer, which is the outermost layer or the epidermal layer, then we can tell that there is an invasion of the pathogen into the tissue space. The mucous membrane do offer a very good kind of first line of defense. All our bodily opening, like our nostrils, our mouth, urinogenital opening, all are lined by membranes, which do contain a mucus layer on top of that. The mucous membrane are often bathed in antimicrobial peptides, antimicrobial enzyme, lysozyme, and they do offer a first line of defense. Then the respiratory tract also is a very powerful first line of defense. So the turbulent airflow deposit microbes onto the mucosal surface. Usually the microbes which are greater than 10 micron are trapped by the hairs and cilia of the nasal cavity. whereas the microbes, which are about less than about 10 micron, pass through the nasal cavity, but they are trapped onto the mucociliary blanket. So let's take a look at what is the mucociliary blanket. So our lower respiratory tract do contain a specific type of cells, and these are columnar epithelial cells, and sometimes known as pseudostratified columnar epithelial cells, and these do contain cilia. On top of these cilia, you have a layer of mucus that we just talked about. So this layer basically traps all these microorganisms which are less than 10 micron and pass through the nasal cavity. So then this layer of cilia do beat from the lower part towards the upper part in a uniform fashion. And this uniform fashion movement is known as the mucociliary escalator. So these transport all these microorganisms away from the lungs, and they can often expelled by the coughing and sneezing or washed out to the stomach saliva. Now, if the mucociliary escalator is breached and the pathogen can enter inside the lungs, there are also some phagocytic cells in the lungs which will take care of that, which will come back later. On the other hand, the gastrointestinal tract do offer some very specific type of chemical defenses. So far, we talked about the physical defenses like our skin or our mucociliary escalator. Those are the barriers. Now we're talking about chemical defenses. What are the chemical defenses? They may contain hydrochloric acid. The stomach contains pH 1 or 2, very acidic, and these hydrochloric acid can kill a lot of pathogen. Also, they do contain the proteolytic enzymes, which can digest pathogen. And of course, there is mucus lining all throughout the gastrointestinal tract as well. On the other hand, the intestine produces pancreatic enzyme BIOL, which will help bind to the lipid, degrade them. Also do contain the intestinal enzyme. There is something which is called gut-associated lymphoid tissue, or GALT. We're going to talk about that. And also the peristalsis is the motion of the intestine by the help of the smooth muscle, which moves the food particle down the intestine. And the process of defecation also can expel microorganisms. Along with that, the shredding of the columnar epithelial cells constantly goes on. So if some of the microbes are attached onto some of the columnar epithelial cells, the flushing activity or the disposal of the columnar epithelial cells can also get rid of them. Also, the normal microbiota is essential for the health of the gastrointestinal tract. These are also the defense mechanism. And why this is important? Because if we lose this normal microbiota, we can get a rise in a certain type of microbes in our body, which can lead to infections. So next is the genitourinary tract and the unfavorable environment for most of the foreign microbes, and the flushing action of the urine, low pH of the urine, and the presence of urea and some of the other metabolite end products in the urine are also antimicrobial. Also, there is another important thing, that the distance barrier in the male urethra do reduce the amount of urinary tract infection or bladder infection. Whereas the innate defense of the vagina is a little bit different because of the closer proximity of the urethra compared to the bladder. Instead of the male urethra is about 20 centimeter compared to in the female it's about 5 centimeter. There are a little bit more chances for the females to get urinary tract or infection or bladder infection compared to a male. But also along with that, there are normal microbiota, which is dominated by the lactobacillus species in the non-pregnant women, which lowers the pH of the urinogenital tract. And that also prevents the growth of many of these pathogens. And also, of course, the mucous membrane, which is rich in lysozyme, many of the antimicrobial defense proteins, as well as we are going to talk about that there are some antibodies in there, but that those antibodies are the part of your adaptive immune response. We're going to talk about those in a later part. But all of these protect the bodily opening from the invasion of the microbes. Then we are looking at some of the chemical mediators, And we just saw that some of these specific areas, like say, for example, genitourinary tract, gastrointestinal tract, respiratory system, mucous membrane, and the skin, these are all different physical barriers as well. And they do secrete some of the chemicals which are responsible for the resistance. And now we are going to talk about that what these chemicals are. So the mammalian host and the normal microbiota produce chemicals to protect against the microbial invasion. Lysozyme hydrolyzes the bond connecting the sugars in the peptidoglycan. So you can see that this is N-acetyl glucosamine and N-acetyl muramic acid, and they are linked by a beta-1,4 linkage. And the lysozyme breaks that bond, so that means the microbial peptidoglycan layer will be dissolved. Once the cell wall of the bacteria are damaged, they will be subjected to osmotic lysis and they can be killed instantly. On the other hand, lactoferrin is found in the breast milk and the mucus secretion. We talked about this before. And similar to transferrin of the blood, this sequester iron means that they limit the ability for the invading microbes to multiply by capturing free iron available to them. Lactoperoxidase, we also talked about that. They also do produce superoxide radicals, which is toxic to many microbes. Along with that, we talked about that there are many different types of proteins and peptides which do have antimicrobial activity. So the first groups are linear alpha helical peptides, and the group is named as cathepsilidins, produced by a variety of cells. The second group are known as the defensin molecules, and these defensins are found in alpha defensins, which are found in the neutrophil, which is a white blood cell. So we're going to talk about that. They are found in an intestinal Paneth cells and intestinal respiratory epithelial cells, and they assist in the bacterial degradation. On the other hand, the beta defenses are found in the epithelial cell, and they stimulate the release of the chemical mediators. That means the beta defensin is responsible to release some secondary chemical signals which will alert the immune system. So alpha defensin is direct degradation of many of the pathogens, whereas the beta defensin alert the immune system for subsequent different activation of the immune system. The third group are known as the histidins, or the larger peptide found in the saliva. They compromise the mitochondrial function and generate the oxidative stress and osmotic stress and result in the fungal death. Bacteriocins are peptides producing by normal microbiota and lethal to the related species. So the difference in between an antibiotic and the bacteriocins are that bacteriocins are usually used in very closely related type of bacteria. They are both produced by the gram-positive or gram-negative cells. For an example, the colicins are produced by the E. coli. So there are some specific type of E. coli which are present as a microbiota in our gastrointestinal tract. They produce vitamins, they are beneficial for us, and they get the food material and help us digesting food. On the other hand, these do produce colicins which will inhibit the growth of some of the non-favorable E. coli or close species. On the other hand, there are some specific streptococcus or bacillus or lactococcus staphylococcus produce some of the lantibiotics. These lantibiotics are bacteriocins, which works the same way, that they do not allow closer species to grow in the gastrointestinal tract mainly. And the reason for that the ones which are beneficial for us are allowed to grow, and that's how the evolution happened along with the host microbiota, which allows a beneficial microorganism to grow, and these lantibiotics or the bacteriocins do not allow the close but pathogenic groups to grow on the skin surface or in the gastrointestinal tract. The next type of chemical mediators are the complement system. Now, this complement system is made up of more than 30 heat labile serum proteins. And these serum proteins circulate in your blood vascular system all the time, and most of them are in an inactive state. So what happens that the complement system have antimicrobial activity, and once they are activated, they convert some of these heat labile serum proteins into the active form from the inactive form, and that allows them to work properly. They are present in the serum all the time, but in an inactive form. Upon a microbial invasion, they can be active. So what they do? They do three major activities. They stimulate an inflammatory response by promoting the recruitment of white blood cells or the leukocyte. So what is an inflammatory response? An inflammatory response is a release of some chemical mediators which do promote a couple of different things. They do increase the blood circulation to a particular site. They can cause this pain and the swelling. That means it is alerting the immune system to fight that specific incidence, which could be due to a pathogen or due to an injury or a tissue stress. But any of these inflammatory response are mediated by the leukocyte or the white blood cells. And the activation of that is also induced by this complement system, along with the chemicals that we have seen before. They also penetrate the microbial cell membrane, causing the cell lysis, and defend against the microbial invasion by promoting the phagocytosis. And this promotion of this phagocytosis is known as opsonization. So there are some specific phagocytic cells in our body which are known as a professional phagocyte. We'll come back to that later. Now, once these complement system do coat them by particular things, the amount of phagocytosis will increase. And this amount of phagocytosis increase is known as opsonization. The process becomes more efficient. So let's take a look at what it does. So here we are looking at the definition of opsonization. So suppose this is a phagocytic cell, and we will come to that, what kind of cells they are. Usually they are macrophages or the neutrophil. And here you have a microbe. And if this microbe is coated by an antibody is secreted by some specific type of cell of our adaptive immune response. We're going to talk about this mainly in the next slide. But if an antibody finds a microbe, it binds onto the surface, and this is the stalk, or we call this one as an FC, that can be recognized by a phagocytic cell, and this will lead to the phagocytosis of this microbe. So degree of binding is just plus. Opsonin is antibody. That means antibody binding to the microbe enhance the rate of phagocytosis. Now, here is another protein, which is a complement protein, and this complement protein is known as C3b. So we are going to talk about that, what is C3b in a later, but think about that the C3b can insert itself onto a microbial surface. Once it does that, this C3b protein on a microbial surface can be recognized by a C3b receptor present on a phagocytic cell. When this interaction happens, it also gives a higher degree of binding, and this will help to phagocytose this microbe faster. So the degree of opsonization is increased compared to the antibody by the binding of the C3b to the C3b receptor of the phagocytic cells. The next one is a double binding, where a microbe is coated by C3b as well as an antibody, and there are two receptors, an antibody FC receptor and a C3b receptor present on a phagocytic cell are binding both. And at that time, the degree of binding is increased dramatically, and the opsonization amount or the degree of phagocytosis will be most effective in this condition. So this just shows the relationship or the degree of opsonization by the phagocytic cells. So next we are looking at that how this complement is activated. We already talked about that the complement is a very complex system which contains more than 30 proteins. And you are going to see that in your textbook, there is a big list of all these different proteins. You do not have to go to each and individual one of them. You just need to know the bigger picture of the complement activation. So the complement can be activated by three different types of pathways. One is a classical pathway, one is a lectin pathway, and another is an alternative pathway. And we're going to talk about that the classical pathway happens if there is a microbe which is bound to antibodies that can trigger the classical pathway. On the other hand, if the microbe do contain some specific type of lectins on their surface, and these are usually some of the specific sugars like mannose that can trigger the lectin pathway. On the other hand, the alternative pathway is triggered by some specific type of molecules present on the microbial surface. Once these pathways are triggered, there are a number of steps or a cascade of reactions which lead to one enzyme to the other, to another protein, to another protein, to another. And after these, they all meet at a particular enzyme which is known as a C3 convertase. So all these different pathways converge into the C3 convertase, which cleaves these C3 molecules into C3A, which is a potent activator of inflammation. On the other hand, the C3b enhances phagocytosis. We saw that in the last slide. So along with C3a, C3b, C3b also activates C5, which activates the C5b production, which produces a membrane attack complex by recruiting C6, 7, 8, 9 molecules, and this makes holes onto the bacterial plasma membrane. Whereas the C5a is also a mediator of inflammation. So there are three outcomes of the complement system. One is phagocytosis, another is a membrane attack complex, another is inflammatory. So let's take a look at how the alternative complement pathway works. This is involved in non-specific defenses, again, intravascular invasion of bacteria or fungus. And it's initiated in response to the bacterial molecules with repetitive structure, for example, lipopolysaccharides. And again, it begins with the activation of the C3 convertase that we have seen, and which results into the formation of the membrane attack complex. We have seen that the C3, on the other hand, activates the C5 that recruits the C5b. And then finally, there are these C5b 6, 7, 8 complexes, which recruits multiple number of these blue or the C9 complexes to make a structure which looks like a hole. And these holes are inserted into the bacterial membrane, and the bacterial membrane becomes completely porous, and you can see that these are the holes on the bacterial membrane, and all the cellular content will leak out, resulting in the microbial death. So this is how the membrane attack complex or MAC lyse bacterial cells. On the other hand, the lectin complement pathway begins with the activation of the C3 and lectin binding, and dependent on interaction of the host mannose binding protein with pathogen surface that we have already seen. So what we mean by that if the pathogen do contain some of the specific carbohydrates like mannose, the host do contain some mannose binding proteins, and this interaction will lead to the activation of the lectin complement pathway. On the other hand, we have already seen the classical complement pathway is dependent on the antigen-antibody interaction and activated when the pathogen persists or when the host is invaded a second time. So these are the different pathways which activate the complement system. The next, we're going to talk about the cytokines. These are soluble proteins or glycoproteins which are released by one cell population that acts as intercellular mediator or signaling molecule. The categories include regulators of innate immunity, regulators of adaptive immunity, and stimulators for hematopoiesis or the production of blood cells. And here, all the hematopoiesis means the production of red as well as white blood cells, but specifically the cytokines or the cellular mediators will activate the white blood cell production, which are the mediators of the immune system. Now, there are several functional groups. The chemokine stimulate the cell migration to a site where there is an invasion of pathogen or there is a tissue injury or stress. Interleukin is another group, is produced by one leukocyte to act another to send a specific signal to do some specific function. We are going to talk about them in detail a little bit later. Interferons, on the other hand, are regulatory cytokine produced in response to an infection, and classically the interferons are related to viral infections. When a specific cell is infected by a virus, these interferons are released, which essentially triggers off a series of response, or they act as transcription factors downstream to produce a lot of different types of proteins which have antimicrobial activity or antiviral activity. Then we have colony stimulating factor or CSF. This stimulates growth and differentiation of immune leukocyte in the bone marrow. Also, there are tumor necrosis factors which stimulate the inflammatory responses, as well as if there is a tissue injury or a tumor in the body like a cancer cells, those can stimulate the inflammatory response as well. So what you are seeing that many of these different cytokines are responsible to activate some of the genes needed to fight a particular condition. And that's the reason that ultimately many of these functional groups, although there are some specific chemokines which stimulate the cell migration or acts as a chemotactic factor, but many of these essentially by step-by-step signaling cause transcriptional activation or turn on transcription of the genes which are responsible for producing many different types of antimicrobial or antipathogenic molecules. Then we have some acute phase proteins. These are present in a macrophage, and we're going to talk about what is macrophage. And these macrophages are a specific type of phagocytic cells present in the tissue and activated by the bacteria, which release a cytokine. And these cytokines, on the other hand, act on the liver, and the liver produce the acute phase protein. So you can easily see that the cytokines are acting on the liver cells and acting as transcription factor to turn on the protein synthesis of these acute phase proteins. The acute phase protein assists in the prevention of the blood loss and ready the host for microbial invasion. These can include the C-reactive protein or CRP, mannose binding protein, we already talked about that, which binds to the different types of carbohydrates on the surface of the bacteria to activate the lectin pathway. There are also surfactant proteins like SPA and the SPD, which also bind and collect on the bacterial surface and acts as opsonin. The C-reactive protein, on the other hand, binds with a specific type of protein called C1q, and the C-reactive protein activates the classical pathway as well. So now we are looking at the cells of the immune systems, and the first thing is the cells of your immune systems are all leukocytes or white blood cells. For example, we know a name of some of them like neutrophils, lymphocyte, monocyte, eosinophil, basophil. They involved in both specific and non-specific immunity, and all arise from a pluripotent stem cells. And we're going to define all these, and each has a specified role. Along with that, they can be grouped as mast cells, granulocytes, monocyte, macrophage, dendritic cells, as well as innate lymphoid cells. This particular slide gives you an overview of the different type of human blood cells. So the first thing is the hematopoietic stem cells in the bone marrow. These are called the pluripotent stem cells, means that these pluripotent stem cells can give rise to many different kinds of blood cells. And these mainly divide into two lineages. One is a common myeloid progenitor cells, which gives rise to erythroblast, which produces the erythrocytes or the red blood cells not needed for our purposes. The second one produces the megakaryocytes, which leads to the platelet. Platelets are involved into the blood clotting and also responsible for inflammation. On the other hand, there are some specific type of unknown precursor that gives rise to the mast cell. So the mast cells are responsible for releasing a specific type of molecule like histamines, which are responsible for allergic reaction. On the other hand, there is a group from this common myeloid progenitor cells, which are called myeloid cells, which give rise to specific type of cells which are known as granulocytes. Why we call them granulocyte? Because when we stain them And under the microscope, light microscope, their cytoplasm looks granular with lots of different granules. That's why we call them granulocytes. So what are these different granulocytes? The first type of granulocyte is eosinophil. The eosinophils are active against fungal as well as helminth or worm infection, and they also mediator from inflammatory pathway. The next one is the basophil, and they are also active in the inflammatory pathway, and their function is sometimes similar to the mast cells, which can lead to allergic reactions as well. Then we have the neutrophil, and the neutrophil is one of the most abundant type of white blood cells or the leukocyte. About 70% of the white blood cells or the leukocyte or the neutrophil. They circulate in the blood, but they leave the blood circulation due to a response to a tissue injury or an invasion of the pathogen into the tissue space. And they are very powerful phagocytic cells, and along with that, they also do secrete a number of different chemicals which are responsible for killing the pathogens. And again, along with these granulocytes, the common myeloid precursor along with the myeloblast also do produce something called, which is known as a monoblast. What is the basic difference in between them? The myeloblasts differentiate into the structures of eosinophil, basophil, or neutrophil, which all of them do have this multi-lobed nucleus. So the nucleus, they are connected by the chromatins, which are the multi-lobed nucleus. Whereas the monoblast differentiate into cells which do have one large nuclei. So the first type of that is the agranulocytes. These agranulocytes do not have any granular structure like the granulocytes in appearance. So this first type, the agranulocyte, is known as the monocyte. These monocytes differentiate from this monoblast, and once these monoblasts produce these monocytes, they circulate into the blood for about 8 hours, and they leave the blood circulation into the tissue space and differentiate into macrophages or the dendritic cells. The macrophages are some of the largest phagocytic cells in the body, and they move into the tissues from place to place. Some of them can remain in the tissues, and they look for pathogens to phagocytose. They also can differentiate into dendritic cells, which also can directly arise from this common myeloid precursor. And these dendritic cells are migratory cells which move around into the tissue, many different tissues, and look for pathogens. Once these dendritic cells do encounter a pathogen, they will capture the pathogen and they will present that pathogen to the immune cells. So that's why we can call them as a sentry cells. On the other hand, these hematopoietic stem cells can also give rise to another, which is called the lymphoid progenitor cells. And the lymphoid progenitor cells can give rise to the lymphocyte, and these lymphocytes can differentiate into, again, another two type of agranulocyte, which are the T cells and the B cells. And we're going to talk about the T cells and the B cells mainly in the adaptive immunity, which is the next chapter. Along with the T cells and the B cells, the common lymphoid progenitor can give rise to the innate lymphoid cells or ILCs, and also the natural killer cells. We are going to talk about the ILCs and the NK cells in this chapter, but we're going to talk about the T cells and the B cells mainly in the next chapter. So first, let's talk about each of these cells. So the mast cells are bone marrow derived cells and differentiate in connective tissues. They contain the granules containing histamine, prostaglandins, serotonins, heparin, dopamine, platelet activating factor, leukotrienes, and all of these influence the tone and the diameter of the blood vessels. What we mean by that, most of these are vasodilators. That means when they release their granules, the dilation of the blood capillaries will increase with increased blood flow. And the vasodilation is often a trigger for inflammation and sometimes the allergic reactions. They also play an important role in allergic responses such as eczema, hay fever, or asthma, where unnecessarily your own immune system is overactivated by some known or unknown reason. It is a little bit of malfunction of your own system where the mast cell release not required some of these chemical molecules to activate your immune system. Say, for example, many of us do suffer from the seasonal allergies from the pollen and the other things. And these are the mast cells which are releasing these different histamine and the prostaglandins to cause this allergic reaction. And when you take particularly the Zyrtex, Claritin, or Allegra and all these anti-allergy medication, they basically bind on top of these specific mast cells and prevent them release of these allergic molecules. The next, the granulocyte, we already talked about that these are the irregularly shaped nuclei with 2 to 5 lobes. The cytoplasm has granules with reactive substances that kill microbes and also enhance inflammation. And basophil, eosinophil, and neutrophils are the different types of granulocyte. So let's take a look at the basophil, irregularly shaped nuclei with usually 2 lobes. And the granules contain histamine, just like the mast cells, and other vasoactive mediators, which always almost increase the diameter of the capillaries or the blood vessels, increasing the blood flow, which cause inflammation. And also they're responsible for allergies and hypersensitivities. Eosinophil is a 2-lobed nucleus connected by a slender thread of chromatin. We just talked about that. And they contain hydrolytic enzyme in addition to peroxidase and major basic protein. So this peroxidase basically will produce some of these superoxide radicals that is responsible for killing the microorganism. They circulate in low number and migrate from the bloodstream to the tissues or like mucous membrane when recruited by chemotactic mediators. What we mean by that, they will remain in the circulation unless they are called in by specific cytokines or specifically some of the chemokines, which are the chemotactic mediators when there is a tissue invasion by some of these pathogens. They also defend against the protozoan and the helminth parasites. Both of them are eukaryotic pathogens. So this is an important definition that eosinophils are responsible for acting against eukaryotic pathogens like protozoan, helminth parasite, as well as fungal infections. They release hydrolytic enzymes, cationic peptides, reactive oxygen species, we just talked about that, and they damage the parasite's plasma membrane, and this is their mode of action. Also, they may play some role in allergic reaction. The neutrophil, on the other hand, has 3 to 5 lobes connected by a slender thread of chromatin. They're highly phagocytic, and they circulate in the blood, and they migrate to the site of the tissue damage. Now, the difference is that the neutrophils mainly will remain in the circulation and they will go into the tissue space when they are being called by mainly chemotactic mediators. That means when there is a tissue injury or if there is invasion of a particular pathogen, at that time the neutrophils will leave the blood circulation rapidly and move into the tissue space. On the other hand, they kill the ingested microbe by hydrolytic enzyme. Also, they produce these reactive oxygen metabolites contained in the primary and the secondary granules. So what happens is that they are phagocytic, and once these pathogens are within the phagocytic vesicles or the phagosomes, inside the phagosome, they will release this hydrolytic enzyme and the reacting oxygen species to kill them intracellularly. They do not release them outside the cell. They are used to kill them after they phagocytose these particular pathogens. So the next one are the monocytes and the macrophages. Remember that the macrophages are highly phagocytic cells, not the monocytes. So the monocytes are the precursor of the macrophages. So the monocytes are produced in the bone marrow and enter the blood, and they circulate for about 8 hours and mature into macrophages and dendritic cells. Now, after maturation into macrophages and the dendritic cells, these will leave blood into the tissue space. And this is a very important concept. Remember that the monocytes are only circulating into the blood for about 8 hours. After that, after the maturation, they leave the blood vessels for the tissue space. And these macrophages are larger than the monocytes as they differentiate, reside in specific tissues, and highly phagocytic and they can migrate and wander around the tissues as well. On the other hand, these act as a sentinel cells sounding the chemical alarm when a microbe invades. That means upon a microbe invasion, they are going to release some of these specific cytokines, which will lead to proliferation of different types of cells as well as different types of chemical reactions. The dendritic cells can arise directly from the lymphoid stem cells, or they can be differentiated from the monocytes just along with the macrophages. They do have these long, slender arms, star-shaped, and they migrate in different tissues and present almost in every tissues. They're present in the skin, mucous membrane of nose, lungs, intestine. They are programmed to detect and phagocytose pathogen. So upon phagocytosis, what they do, they process these foreign antigens which are present on the surface of these pathogens, and they display these information on the surface of themselves. And again, we are going to talk about that how exactly what happens. But just in a summary, upon phagocytosing these pathogens, they display part of the antigens, which could be proteins or glycoproteins, on their surface. These alerts other lymphocytes that there is an invasion going on, and that will lead to secretion of different chemical messengers to alert the immune system. So on the other hand, you can think about that these dendritic cells are also considered as antigen presenting cells. They present an antigen to a variety of different cells to alert the immune system. On the other hand, we just talked about these macrophages as well. The macrophages can also act as antigen presenting cells. On the other hand, the innate lymphoid cells are the common lymphoid progenitor cells, give rise to innate lymphoid cells or the ILCs, including the natural killer cells, and lymphoid tissue inducer cells or LTIs. So these specific cells do not express any antigen-specific receptors, and they lack receptor that responds to remember specific pathogens. So what they do have a different function. The LTI facilitates the development of the tissue found in the lymph node and the Pear's patches. Pear's patches is a specialized type of lymphoid tissue. We're going to talk about that. And these ILCs can be divided into three functional groups. So the first one, ILC1, helps to activate the macrophage when stimulated by intracellular pathogen. That's one of their function, release of cytokines to activate macrophages. Second one, ILC2, triggers vasodilation, release of more mucus and macrophage activity in response to extracellular pathogen such as parasite, also play a role of thermoregulation. That means they can increase the body temperature and can be pyrogenic, or they can lead to fever. The third one, ILC3, is a response to the normal microbiota in addition to microbial pathogens and helps to maintain a barrier function along with the mucous membrane by stimulating release of the antimicrobial peptides, preventing immune response to normal gut microbiota, and promoting phagocytosis and survival of epithelial cells that lines the mucosa. So these are innate lymphoid cells. So not all lymphoid cells are like this, but these are the ILC1, 2, and 3s are responsible for the innate immune response, whereas the specific lymphoid cells or the lymphocytes like T cells and the B cells are mainly the cells of your adaptive immune response that we're going to talk about in the next chapter. So let's take a look in a nutshell that what is happening here. So you have this intracellular pathogen. These are taken care of by the NK cells or the natural killer cells. They kill the infected cell as a whole. On the other hand, the ILC1 activates the macrophages. On the other hand, if there's a parasitic infection, ILC2 increase the mucus production, cause vasodilation, macrophage activation, and also cause thermoregulation. On the other hand, if there are extracellular pathogenic microbiome, right here, the ILC3 can promote phagocytosis, release of antimicrobial peptides, and epithelial cell survival is also triggered in this way. So these are your innate lymphocyte stimuli and responses. So let's talk about the natural killer cells and what they are. These are large granular cells that detect and destroy stressed, malignant, or virally infected cells. They surface the adjacent cells by contacting them via several surface receptors, and if activated to kill the target cell, the NK cells would release pore-forming proteins known as perforin and enzymes called granzymes, and together they will target the cell to commit suicide, which is known as the apoptosis. And also they will attack the cells that are opsonized by antibodies. The process by which the NK cell recognizes and kills such a cell is called antibody-dependent cell-mediated cytotoxicity, or ADCC. So ADCC is a property of the NK cells and a part of the innate immune response, and it is not specific. It could be against any different kind of pathogen. So let's take a look at how NK cells recognize the targets. So although there are some of the specific things which we are going to talk about mainly in the next chapter, but just giving an overview that what happens in a cell once it is infected by a particular pathogen. So here is a target cell, say a normal body cell. And if you carefully look at that on top of this, there are some MHC, an adhesion receptor binding protein. The MHC is known as major histocompatibility complex. And mainly there are two types, MHC1 and MHC2. And all nucleated cells will have MHC1 or major histocompatibility complex 1, whereas the immune cells will have the MHC2s. We're not going to go into the detail of that because that's the discussion of the next chapter. But just in a nutshell, just to understand this process, that if a cell is infected by a particular pathogen, the cell tries to steal at least a couple of these microbial peptides, which could be a viral protein, which could be a bacterial protein, steal them and then show them on the surface of their plasma membrane by the help of this MHC1. What we mean by that? Suppose this is basically signaling or an SOS response by an infected cells. So MHC1 basically shows the flag outside the cell on the plasma membrane, which tells your immune system that I have been infected. And this flag or the microbial peptide or protein is shown by the MHC1. So once this is shown, a particular NK cells or natural killer cells, which are looking for these different types of cells. So these NK cells, which are constantly looking for the cells invaded by the pathogen, will find that there is a particular cell which is showing a microbial protein or a peptide by an MHC1. It will immediately recognize that there is something wrong going on with this cell. It will connect these nanotubes to these targeting cells, and they will do a modification of the cytoskeletal elements, which are mainly the actins or the microfilaments. A cleft will be formed in between the natural killer cell and the target cell. And at that point, into this cleft, this NK cell will release this perforin and the granzymes. These perforin and the granzymes will go into the cell, which will lead to the apoptotic cell death or a programmed cell death. The cell will kill itself. And the reason the cell kills itself, because along with the cell, the pathogen will be killed as well. So this works very well for the virally infected cells. And also the NK cells are responsible for killing tumor cells or cancer cells as well. On the other hand, there is another way that the NK cells can recognize and kill. In here, you can see that there is a typical virus antigen being displayed by an infected cell. And instead of directly producing this type of interaction, before that, suppose an antibody is binding onto the surface of these particular virus antigen. And these virus antigens are showed by the same process by the help of these MHCs or the major histocompatibility complexes. Once these cells, the NK cells, do recognize that there is an antibody being bound on the cell surface, it will immediately come in and release the granzyme and perforins to degrade that. So here is a basic difference in between this type of opsonization by the NK cells and the opsonization by the neutrophil or the macrophages. Remember that here the NK cells, the antibody is bound onto a particular pathogen protein, maybe a virus antigen, on the cell surface where the NK cells is killing the whole cell along with the viruses using these granzyme or perforins. Whereas the opsonization by the macrophages or the neutrophils works in a little bit different way. In those cases, the antibodies do not bind on the cell surface, but the antibodies bind directly on the pathogen, suppose a bacteria. And that bacteria will be phagocytosed by the neutrophil or macrophages, and that is the opsonization. So opsonization does not mean that it is just only the killing of a pathogen by phagocytosis. Opsonization can also include an enhanced killing of the cells by the display of specific antigens by the help of NK cells. So after that, we are looking at the organs and the tissues of the immune systems. And the first thing that we are looking at, the primary organs and the tissues, where lymphocytes mature, differentiate into antigen-sensitive mature B cells and the T cells. And we are also going to look at some of the secondary organs and tissues where the lymphocytes may encounter and bind to the antigen, which triggers their differentiation, activation, and proliferation. So let's take a look at the different tissues. If you look at this, say for an example, tonsils are the lymphoid tissues. Then we have the lymph node, and this is a cross section of these lymph node. Then we have the spleen, and we have these lots of different follicles around our gastrointestinal tracts. We're going to talk about these. Spleen is a very important, which is a tissue of the immune systems. Bone marrow are some of the specific area of primary organs and the tissues. Let's take a look at these individually. So what is primary lymphoid organ and tissues? So let's take a look at the bone marrow first. We will come back to the thymus second. So bone marrow is responsible for producing all of the lymphoid progenitor cells, which will become the T cells and the B cells. Again, repeating, the bone marrow is the only place where all the immature T cells and the B cells are produced. So once they are produced, we are going back to thymus. So the precursor cells, and these precursor cells are the T cells, enter from the bone marrow and proliferate. And what are these precursor cells? These are the precursor T cells. And why we call the T cell? Because the thymus, they mature in the thymus, and the T name comes from the thymus. So the cells that mature in the thymus, the lymphoid cells, are known as the T cells. So here, what are the different things that is going on into these precursor cells? The first thing is called a negative selection. So when these T cells basically develop, they do have a T cell receptor on their surface. These T cell receptors do come in all shapes and sizes to recognize antigens. And these recognition of the antigen could be self-antigen, could be foreign antigen. They are made in a random way with billions of combinations, which can bind to any kind of three-dimensional structure. So once these cells are in the thymus, they undergo a negative selection. What happens there? If any of these T cells, precursor, do react with any of the self-antigen, immediately they will be deleted or they will be killed. So that there is no T cells that's being selected that reacts with our own proteins or self-antigens. The only ones which will be maturing into T cells will not react with any of our proteins or self-antigens. But again, as you know, that this system can go wrong in some of the autoimmune disease where this selection is not 100% correct, and that can lead to autoimmune diseases. So now the remaining ones which are not killed with the negative selection, remaining cells become the mature T cells. They will enter the bloodstream and await activation by the innate immune cells. And particularly, these cells will go into a specific type of tissue, which we known as the secondary lymphoid organs. We're going to see in a second. The next part is the bone marrow also, along with the precursor T cells, do produce the precursor B cells. But the B cells mature in the bone marrow itself. They do not move anywhere else. And now you know that why they are called B cells, because they mature in bone marrow. T cells mature in the thymus, T. B cells mature in the bone marrow. That's why they are B cells. So the maturation involves removal of non-functioning or self-reactive cells the same way. So any of these maturing B cells, if they react with a self-antigen in our body, they will be killed or deleted. Once they are matured, they are only allowed to react with any foreign antigen that is not present in the body. They will enter the bloodstream, will migrate to the lymph node, spleen, and await for the introduction of an antigen, which is a foreign antigen, to which they will make targeted antibodies. So what are the secondary lymphoid organs? Spleen is the most highly organized secondary lymphoid organ. It will filter the blood and it will trap the bloodborne particle for the assessment by the phagocytes and the B cells. The macrophages and the dendritic cells will phagocytose the bloodborne pathogen and will present the antigen to the T cells, which will activate the specific immune response. And again, this will trigger the secondary immune response in the sense that they will activate the adaptive immune response, but we will talk about that in the second part, which is the chapter 33. The other secondary lymphoid organs include the lymph nodes, which also filter the lymph. Microbes and antigen trapped and phagocytosed by macrophages and dendritic cells. On the other hand, the interesting part is that we're going to see that we're going to look at a picture in a second. The outer region is rich in the B cells that can bind to the antigen directly from the blood or lymph. And the inner region is where the T cell await the interaction with the dendritic cells or the B cells. So what does that mean? Although we are going to talk about that in chapter 33, but let's take a look at what's going on in here. So here is a picture of a typical lymph node. And you can see that the outer portion of the lymph node do contain the B cells. The B cells on their surface do contain a B cell receptor, which can bind to the pathogens. On the other hand, the inner area, which is known as a medulla, the outer area is known as a cortex of the lymph node, where you have mainly the B cells. So the cortex contain the B cells, whereas the medulla, which is the inner layer contain the T cells. Now, to activate the T cells, the pathogens do not bind to the T cells directly, unlike B cells. So to activate the T cells, what is needed? That the T cells needed to be presented by the pathogens by any antigen presenting cells, which could be a macrophage, or it could be a dendritic cells, or it could be even a B cells. When a B cells, macrophage, or a dendritic cells or antigen presenting cells present a pathogen or a peptide or a protein of the pathogen to the T cells, it will activate the T cells, which will, on the other hand, trigger the whole of the adaptive immune response. So all these cells from this particular area, which are residing in the lymph node, will rapidly come out, divide, and come out into the bloodstream. That is going to be the topic of the next chapter. So that's what we mean by that. Outer region is rich in the B cells, the cortical area, whereas the inner region of the medulla is rich in the T cells, which are await interaction with the dendritic cells or the B cells. When they capture a pathogen, that is basically when the T cells will be activated. The next type of tissues, which is a secondary lymphoid tissue, are located throughout the body and serves as an interface between the innate and the acquired host immunity, which we are going to talk about in the next chapter. Some lymphoid cells are found closely associated with some specific tissues, and some of these examples are the skin-associated lymphoid tissues, which is known as SALT. On the other hand, there are some other examples like mucus-associated lymphoid tissues or MALT, which includes the GALT, which is gut-associated lymphoid tissue, and the whole of the intestine is covered by this mucus-associated lymphoid tissue. Also the BALT, which is a bronchus-associated lymphoid tissue, which you find in the lungs. And again, remember that all these lung cells are covered by the mucus as well, or the epithelial cells of all of the intestine and the lung passages covered by the mucus-associated lymphoid tissue or the MALT. Now, First, we are looking at the skin-associated lymphoid tissue, and the keratinocytes, which are the skin cells, make up to 90% of the epidermis, and they are capable of secreting cytokines and cause an inflammatory response to invading the pathogen. On the other hand, these Langerhans cells are the specialized dendritic cells which are present in the skin and they wander around. When they encounter an antigen or pathogen, they will phagocytose them and then they will move into the lymphoid tissue where they will present this particular pathogen or the particular antigens associated with the microbe to a T cells, mainly these are helper T cells, which will on the other activate the other T cells and the B cells, which will lead to antibody as well as cytotoxic T cell mediated immunities downstream. Now, these are the part of the adaptive immune response, which is the cytotoxic T cell mediated immunity, which directly kills cells infected with a microorganism, and the B cells, which are going to be differentiated into plasma cells, which are going to secrete the antibodies. But up to this point, it is the part of the SALT or the skin-associated lymphoid tissue where the presentation of this antigen to the T cells happen. Up to that point, it is your innate immunity. After that, the adaptive immunity takes place. The next type is the mucosal-associated lymphoid tissue or the MALT. And here you see the structure of an M cell located between the two epithelial cells in a mucous membrane. The M cells endocytoses pathogen and release it in the pocket containing the helper T cells and the B cells and the macrophages. It is within this pocket that the pathogen is often destroyed. On the other hand, here what you are seeing that the antigen is transported by the M cells to the organized lymphoid follicle containing the B cells, and these activated B cells then mature into the plasma cells, which are producing the antibodies that are released into the lumen, which they react with the antigen that caused their production. And this is the same way that the GALT and the BALT M cells work. So the next part that we are looking at the mechanism of phagocytosis, which is the process the monocytes, dendritic cells, and neutrophil recognize, ingest, and kill cellular microbes. And again, remember that monocytes do not directly engulf or phagocytose. Once the monocytes differentiate into the macrophage, they are responsible for phagocytosis. Now, there are two mechanisms for recognition of microbes by the phagocyte, which is opsonin independent or non-opsonic recognition, whereas opsonin dependent recognition. And phagocytose can be greatly increased by the opsonization. We talked about that with the help of an antibody, with the help of a C3b coat protein, or C3b as well as antibodies both together. So next, what we are looking at that how these opsonin independent pathogen recognition happens. This happens by a specific type of pattern, which is known as a microbe-associated molecular pattern or MAPS, which is also known as a PAMP or pathogen-associated molecular pattern. Now, the MAMP or the microbe-associated molecular pattern is a little bit more inclusive. Sometimes your immune system can also recognize the non-pathogenic bacteria and can lead to some specific recognition as well. And the specific regions within common microbial macromolecules, such as lipopolysaccharide, peptidoglycan, fungal cell wall components, and viral nucleic acid, and other microbial structures, which are very specific to the pathogen and not present in the host cell or the human cell, are recognized by this opsonin-independent pathogen recognition. How these work. So one of them is a calcium-dependent lectin receptor. These binds to the fucose, mannose, or glucans, which are the carbohydrates on bacteria, very specific. Say for an example, Mycobacterium, Helicobacter pylori do have a very specific glucans or very specific sugars. On the other hand, Candida, Aspergillus do have specific cell walls. The helminths do have this chitin on their cuticle, and some of the viruses with the glycoprotein, and also along with that N-acetylglucosamine and N-acetylmuramic acid of the peptidoglycan can bind to these lectin receptors. Along with that, there are some capsular polysaccharides, say for example, Streptococcus pneumoniae, Klebsiella. These all can bind to these lectin receptors, and upon binding, they will trigger the cytokine expression to trigger the immune system. The next one is a toll-like receptors, and there are many different types, as many as 16 different types, and they are found in macrophages, dendritic cells, and many other different cell types. They are present either on the plasma membrane or they are present on the endosome. So they can recognize extracellular pathogens which invaded the body space, or they can also look at the cytosolic pathogens which have invaded into the cells. So upon binding on the TLR, extracytoplasmic or cytoplasm activates a variety of downstream transcription factor, and they will ultimately induce a transcription factor, which is NF-kappa B, to induce some of the pro-inflammatory genes, which will produce the specific proteins which are responsible for inflammation. The next one is NOD, which is the nucleotide and oligomerization domain in the cytosol, and they specifically bind to viral RNAs, some bacterial toxin, intracellular bacteria, and also the NOD is responsible for DAMP, or damage-associated molecular pattern. What we mean by this, that sometimes if a tissue or cell undergoes any kind of damage, they can produce excess amount of uric acid or produce heat shock proteins from cell injury and stress. And these all can be picked up by the NOD-like receptors. And they will form something very important, which is known as an inflammasome. This inflammasome activates a specific caspase 1 protein. And this caspase 1 protein is an enzyme which forms interleukin 1 and interleukin 18. So what we mean by this, that interleukin 1 and interleukin 18 both are produced as precursor molecule and are not activated. But once this NOD-like receptor recruits the caspase 1 into the inflammasome, caspase 1 basically is involved in cleaving the precursor of IL-1 and IL-18 into functional interleukin 1 and 18, which will again undergo a series of activation, step by step, a cascade of activation, and will activate many immune function by activating the transcription factors. The next one is a RIG1-like receptors. This is known as a retinoic acid inducible gene 1, and these are responsible for sensing viral RNA. And upon sensing the viral RNA, the RIG1 receptor downstream will activate many of the transcription factor or antiviral transcription factors, which will specifically trigger production of antiviral cytokine response like interferons. The next, what we are looking at, based on the detection by phagocytosis by conserved microbial molecular pattern, molecular structures that occur in patterns, and MAPs are unique to microbes but not present in the host. As we talked about, that they could be lipopolysaccharide of gram-negative bacteria. Lipopolysaccharide are not present in a host. Example, peptidoglycan gram-positive bacteria. Peptidoglycan is very unique to the bacteria. The MAPs are recognized by a pattern recognition receptors. These pattern recognition receptors are known as PRRs, and they are present on the phagocytic cells. And the PRRs, or the pathogen recognition receptors, can work alone or together to trigger phagocytosis. So one of them is a toll-like receptor we already talked about, functions exclusively as signaling receptors, recognize and bind the unique MAMPs of viruses, bacteria, and fungi. Binding triggers evolutionary ensign signal and is communicated to the host cell nucleus, which initiates the host response. So as you can see that the toll-like receptor can be present on the plasma membrane. There are many different types, TLR4, 2, 1, 2, 6, 5s. Then there are TLR7s or 8s on the endosome, TLR3s on the endosome. And upon binding to specific type of molecules, which are the MAMs, it could be LPS lipopolysaccharide, triacylated lipopeptides, diacylated lipoproteins, flagellin, which is present in the flagella of the bacteria. Also, the different types of RNAs, double-stranded RNAs, which are mainly from the viruses, or single-stranded RNAs. Upon binding to all these types of microbe-associated molecular patterns, this TLRs will activate a cascade of reactions with the help of some other proteins like TRIF, TRAM, MID88, these TIRAP, these are all the different proteins which are involved in the pathway. And you can see that it can activate through the IRF3 pathway a transcription factor, which will specifically trigger this interferon beta production. So this is a cytokine that is produced by this specific pathway. So you can see that double-stranded RNA, when recognized by the TLRs, that will trigger this. Also, the CD4 or TLRs, which is LPS, will trigger this IFN beta. On the other hand, you can see that these are some of the TLRs, 2.1 or 2.6, or even TLR7. They will work through the MYD88 or the TIRAP through the IRAC pathway, which will produce this TRAF6, and they will activate either the MAP kinase pathway, which has not been shown. The MAP kinase pathway can also activate a number of transcription factors. Also, the TRAF6 can also activate NF-kappa B, and this can also induce inflammatory cytokines or different chemokines. And these signaling pathways can induce thousands of different protein molecules in the cell for the antimicrobial response of the cell. So the next one, what we are looking at, that the intracellular digestion and the respiratory burst and how this happens, that the first one, what you are looking at, the PRR on the phagocytic cell will recognize the MAMPs on the microbes, and the microbes will be engulfed with the phagosome, and the lysosome will fuse along with them and forming a phagolysosome. Once that phagolysosome is formed, the lysosomal hydrolases, which contains all the different enzymes like the lipases, proteases, nucleases, will digest the bacteria which has been engulfed. Along with that, it will also cause a respiratory burst, which is the production of reactive oxygen species like superoxides or singlet oxygen. It can also produce the reactive nitrogen species. It can produce hypochlorous acid and hydroxyl radical. All these different species or toxic oxygen products is going to help to kill the microorganism. Once the digestion process is done, the debris can exit the phagocyte by exocytosis. Intracellular parasites such as viruses and bacteria that escape the phagosome are captured by free-floating membranes that form the phagophore. So the cytosolic ubiquitin molecules coat the intracellular invaders and their macromolecules such as viral RNA, and these ubiquitinated particles are then surrounded by this phagophore to form what is called as an autophagosome. Like the phagosome that becomes a phagolysosome during phagocytosis, the autophagosome or the autosome fuses with the lysosome as well, and it will digest its content. Both phagolysosome and the autolysosome can have some of the debris donated to the endoplasmic reticulum, where antigenic peptides are further processed and then bind to the major histocompatibility complex, and then they can be trafficked towards the plasma membrane to be displaced outside so that they can be recognized by the immune system cells that we have seen before. And the rest of the material can be recycled and released into the extracellular environment. So the exocytosis is the process by which the neutrophil expel the microbial fragments after they have been digested, and the phagolysosomes unite with the cell membrane, result in extracellular release of the microbial fragment, very straightforward. And the macrophages and the dendritic cells undergo the process which is called as antigen presentation. We talked about that. And this is the move the fragments from the phagolysosome to endoplasmic reticulum, and the peptide fragment component combined with the glycoprotein becoming part of the cell membrane facing the exterior. And this is done or presented by the major histocompatibility complex or MHCs that we are going to talk about in the next chapter. This helps the presentation of the antigen to other immune cell to alert the immune system to recognize that a pathogen invasion has taken place and to alert the adaptive immune response. So the next part, we are going to talk about the inflammation. And inflammation is a process which is a non-specific response to tissue injury and can be caused by a pathogen or from physical trauma. Acute inflammation is the immediate response of a body to injury or cell death. And there are some cardinal signs and some of the definitions. Rubor is called redness of the area. Warmth is calor, the area feels warm. Dolor is a pain, whereas the swelling is a tumor. And altered function is functio elisa, is the altered function of that tissue space. And these are known as some of the cardinal signs. And these cardinal signs are associated with every type of inflammation. So what is acute inflammatory response? So the release of inflammatory mediators from an injured tissue cells initiate a cascade of events which results in the sign of inflammation. And how this actually works. This involves chemical mediators. So when there is a tissue injury or invasion of a pathogen, that will involve the release of some of the chemical mediators. And these chemical mediators can work on the nearby blood capillaries or the blood vessels, which can express some specific molecules, and selectin is one of them. So selectin is a cell adhesion molecule on activated capillary on endothelial cells. We're going to talk about that in detail in a second. Integrins is another type of adhesion receptors on the neutrophil, which can bind to the selectins. So basically, the process is that how the neutrophils can be bound onto the blood capillary cells. And what is the reason for that? We're going to talk about that. The chemotaxins, on the other hand, are the chemotactic factors which are released by the injured cells. And we're going to see that how they will attract the neutrophil and other cells to the tissue injury site. So let's take a look at what is happening in this process of acute inflammatory response in the injury. So here we are seeing that there is a stab, and due to this, there is a wound in the skin. And with this opening, there are some bacteria that entered into that specific area. And this will result in release of some of the chemicals. And also there will be some phagocytes, which will, like tissue macrophages, will come in and they will try to engulf these microbes, and they will all release different types of chemical mediators. So one of the important chemical mediators, they are going to act on the nearby blood vessels. So what you are seeing that right here is a nearby blood vessels, which contain all these neutrophils inside blood capillary moving. So once these cells express the selectins, you can see that there are some specific adhesion molecules, the selectins are present. It will interact with this integrin or the adhesion molecules, you can see, and the selectins will stop these neutrophils which are moving through these blood capillaries onto these endothelial cells. So again, I'm repeating that this process is known as margination. So in a normal cell, which doesn't have any selectins expressed by the endothelial cells, neutrophil will move through these blood capillaries very rapidly. Whereas when there is a tissue injury, due to some of these chemical mediators will allow these endothelial cells to express these selectin molecules, which will bind to these adhesion molecules like integrins on the neutrophils, and it will allow the neutrophils to stop, and it will start rolling closely to the endothelial cells, not rapidly moving through the blood vessel. And this process is known as margination. And at that point, what happens that there are some other chemical mediators that we're going to see in a second that will allow that there is a little bit of tight junction relaxation in between these endothelial cells, through which these neutrophils will squeeze themselves out into the tissue space. This process is known as diapedesis, where the neutrophil which were present in the blood capillary leaving the blood capillary to the tissue space where there is an injury or an invasion of a pathogen. And now the chemical mediators that we have seen right here, the chemotaxins, these chemotaxins are also released from the tissue injury site or where the pathogen invasion taken place. And the extravasation is the process by which these neutrophils will move into the tissue space and move towards that to enhance the phagocytosis. So here we are looking at the tissue injury leads to activation of one of these specific protein, which is known as kallikrein. This kallikrein releases a specific peptide, which is known as a bradykinin. And this bradykinin leads to swelling. So what they do, there could be a fibrin clot may restrict the pathogen movement. What we mean by that, that upon this tissue injury, there could be some blood leakage into that area, and there could be a blood clot formation around that area, and that may lead to block the pathogen to move into other tissue spaces. So this is a fibrin clot. On the other hand, the phagocytes will accumulate in the influent area to destroy the pathogen that we have seen right here. Phagocytes will accumulate in that area, and the bone marrow will be stimulated to release neutrophils and increase the rate of granulocyte production. And all these neutrophils, which are doing this diapedesis and margination, will move into that area, and they will increase the blood leakage into the tissue at the injured site, which will cause the clot formation, bring more phagocyte, and bring more neutrophil to fight the invasion of the pathogen or deal with the tissue injury. On the other hand, a chronic inflammation is a slow process compared to a acute inflammation. So in the slow process, which involves in the formation of new connective tissue, usually may cause permanent tissue damage. And a granuloma is a well-organized mass of cells of proteins resulting from a chronic inflammatory response. And the epithelioid macrophage in this granuloma are contrasted to a large number of normal macrophage surrounding it. So granuloma is a walled-off area formed when the phagocytic cells cannot destroy the pathogens. And these are very common. What happens that many of these Mycobacterium tuberculosis, which cause the TB, do have this mycolic acid coating on top of them, and many times that phagocytosis by the macrophage is not often feasible. And at that time, these granulomas are formed, and these granulomas are easily visualized under an X-ray as some specific spots. And this area is also densely infiltrated by the lymphocytes and macrophages at the site of inflammation. So here it is a complex subject. Of course, we recognize that. And remember that all the different organs, tissues, and the cells that are responsible for making the first and the second level of defense in this particular chapter. So in this particular chapter, we talked about the basic immunity, which is also known as the innate immunity. And first, you try to learn that what are the different physical barriers. Then the second part, try to learn that what are the chemical barriers. Once this physical or the chemical barriers are reached, which is the first line of defense, then the second line of defense contain the cells of your innate immune system, which involves the macrophages, the neutrophils, different ILCs, and the mast cells, and so on. And we have already talked about in some of the areas that the other side of the coin is the adaptive immunity. And we talked about that some of these T cells and the B cells, how they can be activated. And if it does not make too much sense in here, as soon as you go into the next chapter, it will make much more sense, and you will be able to correlate these two chapters together.